This is the last video for the topic 9.1 segment, and I'm sorry, it will probably be the longest as well. Looking at redox titrations, and there you go, the term titration. We are going back to acids and bases and the concepts that occur in acids and bases, and now applying it with redox reactions. So just like with acids and bases, the purpose of a titration allows us to determine an unknown concentration of a substance in a known solution. And when you reach the equivalence point, just like you would do in an acid-base titration, that is the point in which the reaction taking place has stoichiometrically met the transfers of electrons that will occur. Looking and comparing between the two, acid-base titration versus a redox reaction. So in acid-base reaction, you're neutralizing or running a neutralization reaction between your acid and base. In a redox reaction, you're performing a reaction between an oxidizing agent and a reducing agent. And oddly enough, they can be one and the same. You're just looking at the data from two different lenses, depending on the type of reaction you're running, which is pretty cool. Acid-base, you're specifically looking at the proton transfer from an acid to a base. And if you go back and look at the definitions of oxidation and reduction, it does talk about the gain and loss of hydrogens. And in biology, we tend to look at hydrogen movement to identify redox reactions because it's easier. Chemistry, we focus on the electron side. So that is why you can do an acid-base titration and still look at it as a redox titration, because hydrogen can be used as an identifier in a redox reaction. In a redox reaction, an electron is being transferred from the reducing agent, therefore that guy's being oxidized, to an oxidizing agent who's being therefore reduced, who are gaining the electrons. Same method occurs in a redox titration as an acid-base titration. So you're going to have a burette or a pipette, something that can measure your volume against a standard solution. So our standard solution is always going to be, our standard solution is going to be in the burette. That is what we know. Our unknown solution is going to be in the flask below. And so we're going to go through the same titration method of keep adding a volume of our known into our unknown until we look for that equivalence point. And there's going to be an indicator. Now we may have to add an indicator, or the reaction itself may turn the indicator. So like in this example, MnO4 naturally is purple, as you can see in the burette is purple. And so once you've crested that equivalence point, your solution turns purple, therefore that's your indicator. So depending on, you have to know what kind of reaction you're running to know whether or not you need to add an indicator to the solution. From the volume of the titer used, so what was in the burette, you can therefore find out the concentration of your unknown through the use of the Haack equations. And that's where we're going to go through 3 examples that IB wants you to know. The first example we're going to look at is analysis of iron with a manganate 7 ion. So I'm going to read you the problem. All the iron in a 2-gram tablet was dissolved in an acidic solution and converted to F2+. This was then titrated with KMnO4. The titration required 27.50 centimeters cubed of 0.100 moles per decimeter cubed of KMnO4. Calculate the total mass of iron in the tablet and its percentage by mass. Describe what would be observed during the reaction and how the equivalence point can be detected. This is on page 420 of your textbook if you want to follow along. All right, so looking at what is going to be occurring in this reaction. We are going to be having iron is going from F to So iron is going to be our reducing agent, and it's going from Fe2+ to Fe3+. That's the version of iron in the tablet. Our oxidizing agent is KMnO4. It's going to be in an acidic solution. And so we don't care about the K. That just allows MnO4 to go into solution. So we're going from MnO4 oh my gosh, I can't write, to Mn2+. All right, so then we can go through and we can figure out our full equation of what's going to take place. All right, so just like we did in the second video. So if we look at Fe, Fe is going from +2 to +3. All right, manganese, so oxygen's -2, that's -8. We want to make sure there's a -1 left over, or you can just look at the Roman numeral. It goes from +7 to +2. So we can write our half reactions. All right, so Fe2+ goes to Fe3+. It is being oxidized, so it's losing an electron. So we're going to add 1 electron. All right, so then we have MnO4 negative going to Mn2+. So let's finish out writing this reaction that's occurring in an acidic solution. So now we need to add in our oxygens. We need 4 of them, so 4 waters. So then we need to add in our hydrogens. So our hydrogens, we need 8 of them. And then we need to figure out how many electrons are being transferred. We're going from 7 to 2, so that means we have 5 electrons being transferred. Okay, now make sure your electrons match. We have 1 and we have 5. Well, they don't match, which means we can multiply the iron reaction by 5. So that's going to give us 5 Fe2+, 5 Fe3+, and 5 electrons. Cancel out what's common and write your overall reaction. So 5E2+, oh my gosh, plus MnO4- plus 8H+, gives us 5 Fe3+ plus Mn2+ plus 4 waters. And there we go. We have our stoichiometrically balanced reaction. We need to have this in order to be able to determine the mole ratio of our reactant to our products when we go through the titration method. All right, so now let's do the math. What do we know? We know we're working with a 2-gram tablet that has some iron in it. We know that we use 27.5 centimeters cubed of a 0.100 moles decimeter cubed of K MnO4. Okay, what do we need? Like, what are we looking for? What is our unknown? So it says calculate the total mass of iron in the tablet and its percentage by mass. So we want to know the grams of iron. We're going from MnO4 to Fe. So this is just essentially stoichiometry. So it goes back to topic 1. Everything, whenever we're going between compounds, right, we have to go through the mole ratio. So we need to find moles. So first thing we do, we find moles of our manganate ion that we reacted. So we do this through our concentration calculation, where our concentration times our volume, that's a 2, not a Z, and that's going to give us 0.00275 moles of MnO4 that were reacted during the titration. So since we know our mole ratio of MnO4- to Fe2+, that is a 1:5 ratio. So let's figure that out. So now we need to know moles of iron. And so that is going to take our answer, 0.00275 moles of manganate, multiply that by 5 to find out moles of iron, and we get 0.01375 moles of Fe2+. So if we know moles of Fe2+, we can find mass, right? So moles equals the mass of what we have, like what we're looking for, over molar mass. So the mass of iron that is in that we have that reacted, so we take 0.01375, react that by the molar mass of iron, 55.85 grams per mole, and you get 0.768 grams. So that is how many grams are in the tablet. Now, how much of that tablet is actually iron? So then we take that 0.768 divided by the 2 grams that's in the tablet, multiply it by 100, and you get 38.4%. Observations we may have seen. Okay, MnO4 is purple in the burette, and the flask gets colorless until you hit the equivalence point in which the flask becomes purple, as I mentioned in the previous slide. So that's one example of the titration. Then, looking at the second example IB wants you to know. It's called an iodine thiosulfate reaction. It is on page 422 of your textbook if you want to look at it. So household bleach is an oxidizing agent that contains sodium hypochlorite, NaOCl, as the active ingredient. It reacts with iodide ions in an acidic solution. A 10 centimeter cube sample of bleach was reacted with a solution of excess iodide ions, and the liberated iodine was then titrated with sodium thiosulfate. The titration required 38.65 centimeters cubed of 0.0200 moles per decimeter cubed of the sodium thiosulfate. Determine the concentration of the original bleach used. There's a lot here. Okay. So if we look, and we just kind of some basic information about iodine. Okay, so iodine, when it's in an aqueous solution, will react with an oxidizing agent. That oxidizing agent can be KMnO4, like we used last time, KiO3, K2C2O7, or NaOCl, which is what we're using in this example. So any of these work. And remember, the metals are there to allow for the full dissolving or dissociation of our ions. This is going to provide iodine in its natural form and whatever that reduced product is going to be. Okay? And then iodine gives us a characteristic look. So in this specific instance of where the reaction we care about is taking place. So the titration, what's occurring during the titration, and what we're looking at in the titration. So we're reacting iodine ions with the oxidizing agent, and that's producing iodine and the reduced product. But then we take that iodine and we're going to react it with thiosulfate in the titration. All right, so thiosulfate is going to be oxidized in the titration. Iodine is going to be reduced in the titration. So if we put these two together, we can look at what's going to happen. Okay, so we have our thiosulfate. And the purpose of the thiosulfate is just to react with iodine to figure out how much iodine is in that solution. And I'll show you as we go through this. Okay, So here's the titration that's going to be taking place. So originally, we're reacting iodine ions with sodium hypochlorite, and that's going to produce iodine, but we don't know how much. We're then going to take that iodine and react it with sodium thiosulfate, or the thiosulfate ions. That turns iodine back into its ion form. And this is then going to allow us to figure out how much iodine was present in the original reaction. Now, pure iodine is a deep blue. It actually looks black in concentration. And in its ion form, it's colorless. So we're able to figure out, once it changes from the deep blue to the colorless, how many of those original iodine ions were present in the original reaction that took place, which is kind of cool. So what do we know? We originally worked with 10 centimeters cubed of bleach in our reaction. Then we ran our titration, and we used 38.65 centimeters cubed of a 0.0200 moles per decimeter cubed of your thiosulfate. So just like before, we want to go from thiosulfate to iodine. And then we're going to be able to go from iodine back to the original reaction with the sodium hypochlorite, or the bleach. All right, that means mole ratios. We're always going to the mole. When in doubt, go to the mole. So let's do that. So we want to know how many moles of the thiosulfate reacted in this titration. So we know we had 0.020 multiplied by the volume in which we used. And we are going to find that 7.73 times 10 to the negative 4 moles of thiosulfate was reacted during this titration. So how much iodine was present during that reaction? So then we go to our mole map, or our mole ratio. So our ratio of thiosulfate ions to iodine is at 2 to 1. So that means how much iodine was present? So we take that 7.73 times 10 to the negative 4, and there's 2 to 1, so we need to divide this by 2 to get 3.86 times 10 to the negative 4 moles of iodine. So now we know how much moles of iodine was in the titration. So now we know this was a product of the original reaction. Since this was a product of the original reaction, and you can go back and look at, you had 2 iodine ions reacting with OCl ions. So OCl negative has a negative 1 charge. Then iodine, it occurred in a 1 to 1 ratio. So that means we can figure out how many moles of OCl was in the original reaction. Because it was 1 to 1, it equals the moles of iodine. So it is also 3.865 times 10 to the negative 4 moles of OCl. What is the concentration of bleach that we used? So concentration equals moles over volume. So then we take 3.865 times 10 to the negative 4 divided by the volume of bleach that was originally in that reaction. Remember, it needs to be in decimeters cubed. Equals 0.0387 moles per decimeter cubed. And that, ladies and gentlemen, is how you go. Now, please notice, we're always going to the moles. So make sure you write a balanced reaction so that you can go from the moles. Identify who's the titrate. Identify the volume and concentration of the titrate. Then you go to moles. Then go moles to the other compound that we're looking for. That may be it depending on the reaction. But if there's 2 reactions, now you take the moles of that new compound and take it to the other reaction, so on and so forth. It's not that complicated. Just a lot of small steps. Make a map if you need to of where you need to go. The third one is called the Winkler method. And this calculates what's called dissolved oxygen. So dissolved oxygen, quite literally is the name, is the amount of oxygen dissolved in water. It is an equitable measurement to kind of give us an idea of what the aquatic life is present and how healthy an ecosystem, an aquatic ecosystem is. So by looking at how much DO is present, it allows us to measure the effect of pollution based on what's called the BOD, the biological oxygen demand levels in the waterways. So what happens is if you have a high BOD in your water, that means there's low oxygen levels because that means there's a large biological oxygen demand. There's a lot of organisms who need a lot of oxygen. This is usually caused by nitrates or phosphates present in water, and this process is called eutrophication. So looking at our image, an oligotrophic ecosystem is a healthy ecosystem. It's low in nutrients, so they're limited by nutrients, but this allows for a lot of variety in life. Now let's put in nutrients like nitrates and phosphates, which a lot of that comes from runoff from fertilizers and cleaners in your homes and stuff like that. So that gets into the waterways. Well, what happens is those nitrates allow for single-celled algae cells to proliferate, right? And then they only have about a 24-hour lifespan, so they're going to start to die. Well, that provides food to bacteria, and bacteria consume a lot of oxygen. And so they're going to break down that algae. So no longer can light get in for photosynthesis down here, but also the bacteria is consuming the oxygen present. And that creates what's called the eutrophic system, where there's too much nutrients, so too much bacteria, too little oxygen, death of the ecosystem. And so they use the Winkler method to figure out, well, what do the dissolved oxygen numbers look like so that you can look at what the health of the ecosystem is. It is used worldwide. This is probably the most common form of a redox titration method. Okay. So how we go through this process. First thing we want to do is we want to go through a process called fixing. So we want to fix our O2 with our manganese ion. So we go from manganese 2 to manganese 4 in a basic solution. What this looks like is we're going to have 2 Mn, 2+, react that with the oxygen that would be in the water, and make sure you have a basic solution. That is going to create 2 MnO4 and 2 H2Os. So we have fixed the oxygen. So we can then go through and identify it. So once we fix the oxygen, we want to add what's called acidified iodine. So we fixed our oxygen in the manganate ion. All right, so we are going to go through and we're going to take that manganese dioxide. We're going to react it with the acidified iodine. And that is going to release that manganate ion, make I2 and water. This is starting to look very familiar. This is the iodine thiosulfate titration. So the only thing we changed was the first step, fixing oxygen. So then we're going to titrate that iodine with sodium thiosulfate, just like we did previously. This is a very common method, the iodine thiosulfate titration. It is used all the time. So then we do the same reaction we did previously with the iodine and the sodium thiosulfate. And we're going to create an iodine ion and S4O6, right? So we know, remember, this is a deep blue going to colorless. So there is the equivalence point, right? So now, just so you understand, for every mole of oxygen, we have 4 moles of thiosulfate being used. Okay? Because we have 2 being made while fixing, and so the rest of this needs to be multiplied by 2 because we only reacted 1 mole of MnO2, which we actually would need to react 2 for the amount of moles made in part 1. So keep that in mind. So even though it says, oh, we only use 2 in step 3, it is actually 4 because we have 2 MnO2s. Alrighty, so if we're going to run a problem, what do we know? We have 500 centimeters cubed of a water sample from a waterway. We go through, we fix it with Mn2+, then we use acidified iodine, and then we're going to titrate it with thiosulfate. When we titrate it, we find that we use 12.50 centimeters cubed of a 0.0500 moles per decimeter cubed of Na2S2O3. What is the amount of DO in the water? Okay, guess what? First step, go to moles, because that's the only way we can communicate between our compounds. So just like before, so our concentration 0.0500, multiply that by the volume we used, and we're going to get the moles that were reacted with, 6.25 times 10 to the negative 4 moles. Then do our mole ratio of S2O3 to the amount of iodine. As we saw before, that is a 2-to-1 ratio. So 6.25 times 10 to the negative 4, divide that by 2, and you get 3.175 times 10 to the negative 4 moles of iodine. Then, if we know how much iodine, we're now looking at step 2, like the reaction 2 in the previous slide. So, we know how much MnO2 and the ratio of that to iodine. It's a 1-to-1, so we've gone from reaction 3, this is reaction 2, and if you notice between reaction 3 and reaction 2, iodine is our common compound. Okay, so it's a 1-to-1 ratio, so the moles of MnO2 is the same, so 3.175 times 10 to the negative 4 moles of MnO2. Then we can go back to the original equation in which we fixed O2, and so you can see this is reaction 1, and MnO2 is our commonality between reaction 1 and reaction 2. And we can go through and look at that mole ratio. It is a 2-to-1 ratio. And so we take our 3.175 times 10 to the negative 4, divide that by 2, and you get 1.5875 times 10 to the negative 4 moles of O2. So then what is the mass of oxygen in that water, or what is the mass of oxygen in the amount of moles that have reacted? So that is moles times molar mass. And remember, oxygen is a Brinkelhoff. So then we take that 1.5875 times 10 to the negative 4th, multiply it by 32.00 grams per mole, and that's going to give you 5.080 times 10 to the negative 3 grams in 500 centimeters cubed of water. But if you look here, we are looking for density, so grams per decimeter cubed. So the amount of dissolved oxygen, so you have your grams, change this to decimeters cubed, divide the two, right? So 5.080 times 10 to the negative 3. Divide that by 0.5. And that gives us 0.102 grams per decimeter cubed. And so based off mass per volume or density, there's 0.0102 grams per decimeter cubed of dissolved oxygen. Then you can plot that on a graph and you can look at it over time. You can compare it to other locations. You can look at concentration of nitrates and phosphates and how much dissolved oxygen. There's a lot of things we can then use this information for to track the health of an ecosystem. So there are 3 types of titrations, but if you look in example 2 and example 3, a lot of it is the same. A lot of it's just going back and forth between mole ratios. So really pay attention to what it is you're reacting in the titrate and what it is you are looking for, so you know how many mole ratios you have to go through to get your final answer.